So welcome to the first Neuroscience lecture for 2020. For this lecture, I'll start by taking you through a brief introduction and history of neuroscience before running you through some anatomy and finishing up on the more accessible topic of membrane potentials. So to start, neuroscience is essentially the study of the structure and function of the nervous system, and it ranges from cellular and molecular biology to gross anatomy, physiology, disease states, behavior, and even cognition. Through it, we've come to learn that the brain governs various aspects of human nature and functions such as pain, emotion, and mood. So modern neuroscience took some time to achieve, but what we see throughout history is that we have evidence of neuroscientific ventures. For example, we see early forms of neurosurgeries via trepanation, where our early ancestors essentially drilled or scraped a hole into the skull. And this is what we assume was to try and treat health problems, but we're still not 100% sure as to why this was done. Mind you, when we look through history, the brain actually wasn't the always go-to organ. The ancient Egyptians actually believed that the heart was where our soul and memories were tied. So plenty of pseudoscience preceded our current understanding of modern-day neuroscience. A great example of that is phrenology. This was created by Franz Joseph Gall, and this was around the 18th century. He believed that the size and shape of the skull, including all the little ridges and bumps on the surface of the skull, reflected personality traits, and he therefore correlated structure of and shape of the skull to personality and behavioral traits. However, this theory itself wasn't popular for long, as it was proven to be completely incorrect. We then moved through the 19th century, where there was significant progress in the area. We started to recognize several things, such as that the nervous system was able to generate electricity, and that nerves essentially acted as wires to conduct that electricity. We also recognized that different parts of the brain were able to perform specific functions. For example, we found that there was a specific area for speech, known as the Broca's area, and that the occipital lobe was important for vision, and that the cerebellum played crucial roles in movement and coordination, and so on. We also recognized that neurons were the basic functional unit of communication. From these progressions, we now have modern neuroscience that we use today. So that's a little bit of history. Now, to gain an understanding of neuronal functioning and disease states, you need to have a basic idea about neurons and neuroanatomy. So first up, when discussing the nervous system and the brain, you'll remember that it's packed with nervous tissue, very little of which is made up of extracellular space. Most of that tissue is actually made up of cells, the most abundant and important of those cells being your glial cells, also known as neuroglia, and your neurons. Glial cells are the most abundant type of cell in the brain, making up about half of all your brain cells. They essentially support neuronal function, so they insulate and nourish neurons. Our neurons are nerve cells that transmit signals via electrochemical changes in their membrane. So translation, they respond to stimuli and send and receive signals. These are your most important cells in the central nervous system because neurons are how the brain communicates, so they are the backbone for normal functioning. Interestingly, most central nervous system neurons actually lack a structure called a centriole and therefore cannot divide. And consequently, this is why neurons that are lost to injury or disease are seldom replaced in the central nervous system. When we look at a prototypical neuron, we see that neurons have a cytoskeleton made up of microtubules, microfilaments, and neurofilaments. The reason I mention the cytoskeleton is because when the cytoskeleton is compromised or disrupted, you get a loss of neuronal functioning, and in turn a loss of brain functioning, which is the basis for neurodegenerative diseases such as Alzheimer's disease. And this is the most common form of dementia, accounting for about 60 to 70% of cases. For example, in Alzheimer's disease, the tangle formation in the cerebral cortex is caused by hypophosphorylation of tau proteins. Tau proteins ensures microtubules run straight. What happens in Alzheimer's disease is that tau protein detaches from microtubules or its functioning gets disrupted, and this causes axons to wither and tangle as the microtubule is no longer able to stay tough and straight. This results in disrupted communication throughout the central nervous system, affecting cognitive functioning. Through this example, we start to see that disruptions in certain components of a neuron can affect basic functioning. We also have other important components, specifically the axon terminal, and the dendrites. The axon is the elongate extension portion that conducts a signal along the length of the neuron. The axon terminal houses the synapse, which is the junction that separates two neurons, and is the spot that actually transmits signals to the next neuron. Dendrites essentially act like the antenna of neurons, detecting signals sent or received from other neighboring neurons. Another easy way to remember dendrites is as finger-like projections. Now, most neurons in the brain will look like this. When a signal is transmitted, it travels from the dendrites down the axon to the axon terminal and synapse. When it reaches the synapse, the signal will be sent from the synapse of that neuron, which we call the presynaptic neuron because it sends the signal, to the dendrite of the next neuron, which we call the postsynaptic neuron because it receives the signal. If you look up close, it looks like this diagram here. You have the synapse of the presynaptic neuron, which sends the signal, and the dendrite of the postsynaptic neuron that receives the signal. So the axon terminal and synapse is where signals get sent from one neuron to the next. When we talk about synaptic transmission, we are talking about sending signals from one neuron to the next via a synapse. Another thing to note about synaptic transmission is that signals go through electrical to chemical to electrical transformation. This is because when it travels through the neuron, the signal is electrical, but when it passes from one neuron to the next, it transfers to a chemical signal. So when the signal gets to the synapse, that neuron will release chemicals, also known as neurotransmitters, that diffuse across a little gap, synaptic cleft, between the two neurons. And those chemicals will bind to the next neuron and the signal will get converted back to an electrical signal. So when we talk about the principal form of signal transmission, it is technically chemical and not electrical. As in the next slide, what you can see is that some neurons can sometimes have a cover that runs over the length of the axon. So you can see the blue parts running over the axon in the top diagram. If you look at these covers close up, it looks like a little like the bottom diagram. There are specific types of myelinating glial cells in both the central nervous system and the peripheral nervous system. In the central nervous system, we have oligodendrocytes, and these cells are responsible for producing myelin. And this is that membranous coating that insulates and surrounds axons and increases the speed of nerve transmission. They also assist in stabilizing portions of axons. In the peripheral nervous system, so i.e. that's all neural tissue outside the brain and the spinal cord, they are known as Schwann cells. What you can also see in the lower diagram is that in between each sheath is a small gap. This exposed region of the axon is known as the node of Ranvier. The major advantage of myelin sheaths is that neuronal signals can actually travel faster when an axon is myelinated, as the signal can jump from node to node rather than traveling through the entire length of the axon. So next up, you'll need to have a general idea about the various branches of the nervous system. The main branches are the central nervous system, which consists of the brain and the spinal cord, and the peripheral nervous system, which consists of all neural tissue outside the central nervous system. The peripheral nervous system can be divided into its autonomic and somatic divisions, which can be further subdivided. The autonomic nervous system consists of two branches, the sympathetic, which is also known as our fight or flight response, and the parasympathetic division, which is known as our rest and digest response. Whereas the somatic nervous system can be divided into both sensory and motor divisions. You can then see an overview of the brain where you can see its three main components of gross anatomical structures, which are the cerebrum, which is the largest part of the brain, the cerebellum, which is the mini brain that's known for coordination and modulation of motor commands, and the brainstem, and this connects the brain and the spinal cord. If you look at the underside of the brain, you can see the two distinct hemispheres, the left and the right hemispheres. However, there exists also a small gap between them, so i.e. they don't necessarily touch. This gap is known as your longitudinal fissure, and the structure that connects the two hemispheres is called the corpus callosum. This is a thick bundle of nerve fibers that connects the two cerebral hemispheres and allows them to communicate. Moving on, we again see the cerebrum, also known as the cerebral cortex, but we now have it divided into its respective lobes. There are four main lobes: the frontal, parietal, temporal, and occipital lobe. You will need to remember the lobes and have a basic idea of some of their functions. For example, the frontal lobe is important for planning and decision making, whereas the parietal lobe is important for processing sensory information. And as mentioned earlier, the occipital lobe plays crucial roles in visual processing, while the temporal lobe contains brain structures that are important for memory, hearing, and emotion. So the cerebrum is also highly folded and contains what appear like wrinkles. These wrinkles consist of gyri, which are the small bumps that you feel on the surface of the brain, and sulci, which are shallow depressions that separate these adjacent gyri. Together, these folds increase the surface area of the brain, allowing a large amount of tissue to fit within the skull. I also want you to have a general idea about the meningeal layers that support and protect the brain. There are three main components: the dura mater, the arachnoid mater, and the pia mater. The dura mater is the outermost covering of the brain and consists of two outer layers of tough fibrous matter. The layers of the dura mater form a fold that separates the cerebral hemispheres and forms a second fold that holds the cerebrum up off the cerebellum. You then have the arachnoid mater, which is the middle layer of the meninges, so called because it resembles a spider's web. This layer lies between the dura and pia mater. Underneath the arachnoid is a space known as the subarachnoid space. It is in this space where cerebral spinal fluid flows, and the main function of cerebral spinal fluid is primarily to cushion the brain, but also assist in the transfer and distribution of nutrients to neurons as well. Finally, you have your pia mater, the soft matter that lies right on the surface of the brain and holds the cerebral arteries in place. It is highly vascularized, with blood vessels that perforate through the membrane to supply the underlying neural tissue. Now, the brain also has an important ventricular system. It contains four cavities, or four ventricles, and these each contain cerebral spinal fluid. Your ventricles actually have specialized tissue called the choroid plexus, and this secretes the cerebral spinal fluid. Next up is the blood-brain barrier, which is a complex that surrounds most of the blood vessels in the brain and acts as a protective and selective physiological barrier between the bloodstream and the extracellular space around it. But it's important to note that some brain regions actually don't have a blood-brain barrier. For example, the hypothalamus. This is because it needs to release hormones into the circulation, and it would not be able to achieve this with the barrier, and so it forgoes it. So in summary, it surrounds most but not all of the blood vessels in your brain. The blood-brain barrier regulates the movement of ions, proteins, and other blood-borne substances into the central nervous system, and in turn prevents the unwanted passage of substances into the brain, maintaining the highly regulated environment. This ensures reliable signaling and synaptic functioning within the brain. There are also a couple of central components of the blood-brain barrier that are unique and not found in other blood vessels throughout the body. The first are tight junctions. Normal blood vessels typically have pores that allow for the passage of material, but in the blood-brain barrier, those pores don't exist because the endothelial cells actually fuse together to form tight junctions. The blood-brain barrier also has astrocytic end feet that completely surround the blood vessel, and you don't see astrocytic end feet with normal blood vessels. The astrocytic end feet help to support and nourish the blood-brain barrier. So these are a couple of important components that help make up the blood-brain barrier and are unique to it: the tight junctions and the astrocytic feet, namely. So next up, we then see the cortical areas of the brain. We see our three motor areas: the primary motor cortex, the premotor cortex, and the supplementary cortex. We can also see various sensory regions. For example, the somatosensory cortex, the occipital cortex, which is involved in visual processing, our auditory cortex involved in hearing, and the gustatory cortex, which is involved in gustation. So that's our sense of taste. On this slide, we have our thalamus, and the thalamus acts as a major relay center for both sensory and motor signals. In many ways, the thalamus is like the Australia Post of the brain. It receives signals from different areas and sends these signals to the appropriate brain regions for further processing. We then also have our hypothalamus. The functions of the hypothalamus are diverse and multifactorial, but the main ones you'll need to know is that it acts as a link between the nervous and endocrine systems, and that it controls hormone secretion by the anterior pituitary gland. Moving on to our central part of the brain, we then have our tectum and tegmentum, which together comprise our midbrain. The tectum consists of the superior and inferior colliculus, whereas the tegmentum consists of the substantia nigra, the red nucleus, and the ventral tegmental area. In the lower part of the brainstem, we have the medulla oblongata. This structure plays important roles in regulating autonomic functions, including heart rate, blood pressure, and respiration. Meanwhile, we also have the pons, and this sits slightly above the medulla. This connects the cerebellum to the brainstem and contains nuclei that control hearing and equilibrium, etc. We then have our cerebellum, and the cerebellum is involved in the preparation and modulation of motor commands and control. However, there is now also emerging data suggesting it may be involved in attention and language as well as fear and pleasure. We then have the limbic system divided into a collection of structures. These include the telencephalon, the diencephalon, and the mesencephalon. The main function of the limbic system that you will need to know is that it processes and controls emotional regulation and is associated with emotion, behavior, motivation, and memory. When we look at the spinal cord is attached directly to the brain and is a conduit of information from the brain to the body. You will need to have a general idea about its dorsal and ventral columns, as well as its dorsal and ventral roots and horns. On this slide, we have our cranial nerves, which all serve important physiological functions. You will need to know the 12 pairs of cranial nerves by name and by Roman numeral, as this is how they commonly appear or are assessed in exams. It would also be prudent to have an understanding of their general function. So broadly, the function of cranial nerves can be divided into 4 categories: sensory, motor, special sensory, and both sensory and motor. The olfactory nerve is an example of a special sensory nerve, and this innervates the olfactory epithelium and is involved in the sensation of smell. We then have the optic nerve, cranial nerve number 2. This is a special sensory nerve, and this is responsible for innervation in the retina of the eye, so obviously in seeing things. We then have our oculomotor nerve, which, as the name suggests, is a motor cranial nerve, and this is involved in moving some of the muscles of the eye, for example, the intrinsic eye muscles. We then have our trochlear nerve. This is an example of a motor cranial nerve, and this innervates the superior oblique muscle. We then have the trigeminal nerve, cranial nerve number 5, and so called because it has 3 different divisions. So because of that, the primary function of the trigeminal nerve is mixed, so it's both sensory and motor. We then have our abducens, and this is an example of a motor cranial nerve, and it primarily innervates the lateral rectus muscle. Cranial nerve number 7 is our facial nerve, and its primary function is a mixed cranial nerve. This is because it has both sensory functions, so it innervates the taste receptors on the anterior 2/3 of the tongue, and it also has motor functions, so it innervates the muscles of facial expression, the lacrimal gland, and the sublingual salivary glands. We then have our vestibulocochlear nerve, which is also known as the acoustic or one for hearing, and this is an example of a special sensory cranial nerve. It primarily innervates the cochlea, so that's our receptors for hearing, and it also innervates the vestibule, so that's our receptors for motion and balance. Cranial nerve number 8 is also known as the auditory vestibular cranial nerve. We then have our glossopharyngeal nerve, and its primary function is mixed, so it's both sensory and motor. Its sensory functions include innervating the posterior third of our tongue, the pharynx and the part of the palate, as well as receptors for blood pressure, pH, oxygen, and carbon dioxide concentrations. Its motor functions include innervation of the pharyngeal muscles, as well as the parotid salivary gland. We then have cranial nerve number 10, which is known as the vagus nerve, and this is the longest cranial nerve in the body. Its primary function is mixed, so it has both sensory and motor functions again. Its sensory functions include innervation of the pharynx, the auricle and external acoustic canal, the diaphragm, the visceral organs in the thoracic, as well as the abdominal pelvic cavities. And in terms of its motor functions, it's also important in innervating pharyngeal muscles and visceral organs in the thoracic and abdominal pelvic cavities again. Cranial nerve number 11 is known as our accessory cranial nerve, and this is an example of a motor cranial nerve. So this innervates our skeletal muscles of the palate, the pharynx, and the larynx. It also innervates the sternocleidomastoid and trapezius muscles. Finally, we then have our hypoglossal cranial nerve, and this is an example of a motor cranial nerve because it innervates and controls tongue musculature. Just shifting gears a little bit, we now have some information about the human connectome. Essentially, the human connectome is an ongoing project that aims to provide a comprehensive compilation of neural data to graphically navigate the brain. This is undertaken by various researchers globally and is not confined to the brain, with connectomes being progressively developed for other anatomical structures. Okay, so for the second part of this lecture, we're going to be concentrating on membrane potentials. So minds as electrically excitable, so that's both our muscles and neuronal cells, and non-electrically excitable, which includes our endothelial cells and connective tissue. All of your electrically excitable cells have a negative resting membrane potential. This generally ranges from negative 5 millivolts to 100 millivolts. The average normal resting membrane potential of neurons is usually negative 65 to negative 70 millivolts. The membrane potential is created by the difference in the number of ions inside a cell compared to outside the cell. So to reiterate, the resting membrane potential of a typical neuron is minus 65 to minus 70 millivolts. It is negative because at rest, the inside of the cell is more negative than the outside of the cell. For those of you wondering why it is more negative, well, at rest, there are a larger number positive of sodium ions outside the cell and a small number of positive potassium ions inside the cell. Because the inside of the cell has less positive ions and a large amount of negative ions as well, the inside of the cell is considered more negative compared to the outside of the cell. Now we maintain the resting potential in a few different ways. The first way is with selectively permeable plasma membrane, which separates the contents of the cells from the outside environment. We also have concentration gradients for individual ions that distribute across the plasma membrane. We then also have the presence of ion channels and pumps, namely the sodium-potassium pump, and these ion channels will open or close to move ions in and out of the cell. And last but not least, we also have the presence of leak channels, and these are channels that are constantly open and allow ions to leak in or out of the cell. So again, just to reiterate, the resting membrane potential exists when there is a difference or uneven imbalance in the number of ions inside a cell compared to the outside of the cell. Something to keep in mind is that the cytosol and extracellular fluids are electrically neutral, so they themselves don't influence membrane potentials at all. And it's important to note that the difference in charge applies only to the small distance across the membrane. Now, there are different ways in which the membrane potential can change. Specifically, we have passive mechanisms, so that involves simple diffusion and facilitated diffusion, and we also have active transport, which lets ions diffuse in or out of the cell. Remember that ions are both positive or negative molecules, just like sodium and potassium. Now, the movement of ions is always facilitated by either a channel protein or a carrier protein. Channel proteins are also known as ion channels. These ion channels can open or close to allow ions to pass passively through the plasma membrane down their concentration gradient. How the ion channel opens or closes is known as gating kinetics, and it usually depends on the stimulus. So we have voltage gating. So this means that the ion channel opens or closes in response to changes in the membrane potential. We have chemical or ligand gating, which means that the channel will only open when a specific chemical or neurotransmitter binds to the channel. And lastly, we have mechanical gating, and this relies on a physical change or distortion in the shape of the plasma membrane. Figure 4 just gives you a nice visual representation of gating kinetics. And again, just to reiterate, chemical or ligand gating, this means that the channel will open only when a specific chemical or neurotransmitter binds to the channel. Mechanical gating implies or relies on a physical change or distortion in the shape of the plasma membrane. And then we also have voltage gating, which means that the ion channel will either open or close in response to changes in the membrane potential. Finally, and as mentioned previously, we also have our leak channels. These are constantly open and allow sodium and potassium ions to leak in or out of the cell. So when ions need to be transported against their concentration gradient, we then have the sodium-potassium pump, and this is an example of active transport. The sodium-potassium pump moves ions in and out of the cell, but it uses energy in the form of ATP to do it. The pump maneuvers 3 sodium ions out of the cell for every 2 potassium ions that move into the cell. This results in a net loss of positive charges within the cell. So in summary, it compensates for the leaky channels and it helps maintain the resting membrane potential of the neuron. So resting membrane potentials can also be modeled by an artificial membrane that separates two chambers. As shown in the diagram below, in situation A, we have an impermeable membrane separating two regions, one of high salt concentration and the other of low salt concentration. The relative concentrations are represented by the sizes of the letters. In situation B, inserting a channel that is selectively permeable to potassium results in a net movement of potassium down its concentration gradient, so from left to right. So it moves from an area of high concentration to an area of low concentration until the two areas are equal. In situation C, when equilibrium is established, there is no net movement of ions across the membrane. 7. All I want you to remember is that when equilibrium is reached, there is no net movement of ions in and out of the cell. We can also measure equilibrium potential using a specific equation called the Nernst equation, but we don't expect you to use it. Now, when we send signals in the nervous system, it often involves the generation of electrical current in the form of a change in the membrane potential of a cell. This usually happens when there is an alteration in the ion concentrations across the membrane, and these alterations essentially lead to the opening and closing of ion channels. When these channels open, they allow lots of ions to enter the cell, changing the membrane potential even more. So there are two types of signals that can be produced by changing the membrane potential: graded and action potentials. Both are electrical signals that the brain uses to communicate. So a graded potential occurs when an ion channel allows ions to move in or out of the cell, causing a small change to the resting membrane potential. There are a few key things to remember about graded potentials. One is that they induce small changes in the membrane potential. Two is that the magnitude of a graded potential varies with the strength of the stimulus. Three is that the signal travels over short distances compared to an action potential. And four, being that the signal will eventually decay. Now, the role of graded potentials is to excite, so to depolarize, or to inhibit, also known as hyperpolarization, action potentials. When we talk about ions moving in and out of a cell and that resulting change in membrane potential, we are talking about an increase or decrease in polarity. A decrease in polarity, so i.e. less negative and more positive, is caused by sodium ions entering the cell. This is called depolarization. When we have an increase in polarity, so i.e. becoming more negative, this is caused by potassium ions leaving the cell. This is called hyperpolarization. Depolarization is a reduction in the membrane potential. So for example, a change in the membrane potential from minus 70 millivolts to minus 60 millivolts. And hyperpolarization is an increase in the membrane potential. So that's a change in the membrane potential from minus 70 millivolts to minus 80 millivolts. We also need to note that depolarization does increase the probability of producing nerve impulses, whereas hyperpolarization decreases its probability. Slide 41 is the concept of an action potential. An action potential occurs when a graded potential reaches a specific threshold value, being approximately minus 55 millivolts. Action potentials are the type of signal used for long-distance communication. Now, during an action potential, there is a temporary reversal in the charge of the resting membrane potential. However, unlike a graded potential, the action potential magnitude remains the same along the length of the axon. We call the firing of an action potential the all or nothing principle, because if the graded potential doesn't reach that threshold value, there won't be an action potential. So with respect to the all or nothing principle, there are also a few important components to remember. One is that depolarization must reach a threshold value to generate an action potential. Two, action potential magnitude remains consistent across the length of the axon. Three is that the movement of the action potential triggers the next section of the axon to fire. Four is that various stimuli can induce a change in the membrane potential. And five, all stimuli that bring the membrane to threshold will generate identical action potentials. So simply put, the bottom line is this: if a stimulus does not reach the threshold, which is usually at negative 55 millivolts, there will be no action potential. Only the firing frequency may differ, and this is dependent on the magnitude of the depolarizing current. So here on slide 42, we have a nice visual representation of an action potential. It is important that you understand all components of the action potential. At rest, a cell sits at approximately -65 to -70 millivolts. A stimulus will then come along and cause the cell to depolarize a little. If that depolarizes is sufficient and reaches a specific threshold value of roughly -55 millivolts, it will then generate an action potential. The action potential then has a few distinct phases. First is the rising phase, and this is when you have an influx of sodium ions moving into the cell. This makes the cell more positive, so we're pushing the membrane potential to around +30 millivolts. Subsequently, we then have the falling phase of the action potential, and this is where we have lots of potassium ions leaving the cell, and this makes the membrane potential negative again. But instead of stopping at -70 millivolts, it tends to overshoot a little bit, and we call this phase the hyperpolarization phase. This occurs because of those leak channels of potassium ions. Eventually, the sodium-potassium pump and the leak channels will work together to bring the membrane potential back to -70 millivolts. The other thing to note is that during the rising and falling phase, we cannot generate another action potential because there is a temporary inactivation of the sodium channels. We call this the absolute refractory period, and this lasts for about a millisecond during that rising and falling phase of the action potential. So in summary, during the absolute refractory period, we cannot generate another action potential. We also have a second refractory period, and this is called the relative refractory period. This occurs during the hyperpolarization phase. The relative refractory period lasts a few milliseconds, and during this time, you can generate another action potential, but the stimulus will need to be stronger than the one before it. So these are the phases of an action potential, and as mentioned earlier, it would be wise to understand and be able to label all the individual components of an action potential. Finally, there are two ways that a graded or action potential can move down an axon, these being continuous or saltatory conduction. In continuous conduction, this occurs along unmyelinated axons, and the action potential seems to move in a series of tiny steps. Continuous conduction cannot occur along myelinated axons because myelin blocks the flow of ions across the membrane. Remember, ions can only cross the plasma membrane at the nodes of Ranvier, which, if you recall earlier, refers to the exposed region of an axon. By contrast, in saltatory conduction, depolarization and propagation occurs at the nodes of Ranvier. Remember when I mentioned that signals travel faster on myelinated neurons because the signal can jump from node to node. That's why we call it saltatory conduction, and it occurs much faster than continuous conduction, which occurs at about 1 meter per second. If a neuron isn't myelinated, it will travel down the entire length of the axon, which we call continuous conduction, and this occurs at a rate of approximately 100 meters per second, that being for saltatory conduction. The last thing to remember is that an action potential can only travel one way down an axon, and that there are differences among myelinated axons. This is because the speed of impulse transmission varies with axon diameter. For example, the larger the diameter, the lower the resistance to ion movement, and therefore the faster the current travels. So that's it on the history, neuroanatomy, and membrane potentials. Thank you for tuning in. If you have any questions from today's lecture, feel free to email your lecturer, Shimona Maharaj, who developed the lecture content, at shimona.maharaj@uts.edu.au. Thank you.